So diagrams. Okay. So in the paper 1, test 1 in paper 1, most of the topics are general. Yeah. Okay. So I asked questions like conservation agriculture, sustainable agriculture, precision farming. Okay. So those are the questions in the paper test 1, right? So most of these topics are general in nature. So you know the answer, you can able to answer the question, but the problem is how to structurize those answers. Okay, and how to because most of the time in the paper 1, maximum you can get around 140. That's all. Okay, so crossing 140 and all is very difficult in paper 1. Okay, and paper 2, the questions are highly technical in nature, and recently UPSC asking more specific questions. Okay, the question is not general, so they are asking very specific questions. For example, even within the cell biology. Okay, so see the nature of question. Define cell. How is a plant cell different from animal cell? Describe a typical cell with a suitable diagram. This is a general question. Okay, this one type of question. Okay, so simply it's a overall. So you have to define what is cell. Okay, so cell is the basic unit of life. Okay, cell is the basic structure and function of life. Okay, and then you can differentiate between a plant cell and animal cell, the common differences between plants and animal cell, and then you can write describe the cell with the suitable diagram. So you have to draw the plant cell diagram, and you have to define describe each part of the cell. That's all. Okay. So here it's a 15 marks question, 3 pages. So within 3 pages, you have to contain your answer. So you have to write a short description of each the plant cell parts. Okay, right. Apart from the diagram, neat diagram. Okay, right. It's a general question. But in addition to that, in the same cell structure question, we can expect some very specific questions like describe the ultrastructure of the mitochondria, and describe the structure and function of mitochondria or plastids. Okay, that's why I asked this question. Okay, the plastids. Write short notes on the following plastids. So describe the ultrastructure of ribosomes. Okay, so these are the very specific questions. So you have to read the overall. Okay, suppose if your question is the general question, you have to read overall. You have to. There are a lot of, for example, in the cell question, more than you have to read more than 5-6 pages. Okay, you have to contain the entire answer into 3 pages. At the same time, very specific questions. Okay, for example, the special types of chromosome. Okay, usually we study about the chromosome. Okay, in general, the chromosome structure and the function. Okay, but here the question is about special types of chromosome and what are the genetic significance of chromosome. Okay, such questions are possible. Okay, right. So everything you can. So for class students, you can find answer in the notes, our material. Okay, I have covered even the minute details. Okay, about every topic. Okay, the possible questions. And here one more question. Yeah, the distinguish between monohaploid and the polyhaploids. So this is what I want to. So the recently you have to focus on these things, right? So we know what is haploid, okay, and in particular they are asking about the monohaploid and the polyhaploids. Okay, so these are the very specific questions. They are asking even minute details, okay, minute parts of the particular topic, right? Okay. So this is the nature of the second, okay, the paper 2, right? So the advantage is, if you know the answer, you can easily get very good marks in paper 2, right? So even crossing 160 is a easy task, okay, in paper 2. You can easily cross 160, 165, that is possible. So if you can able to score around 140, 145 in paper 1 and 160 above in paper 2, then you can easily touch 300. Okay, because nowadays the optional in general maximum score in average, maximum score in optional is around 270 to 80 only. Okay, 300 is not a very common score in optional nowadays. Okay, right. So if you touch 300, 290, 300, then definitely you will get a renewal of the service. You'll get an interview call, and based on your performance on the SC and the GS, you'll get a renewal of the service. Okay, right. So that's our target. Okay, so crossing 290. Okay, right. So I will provide a detailed answers for all these questions. Okay, and you can also refer our notes, the class notes. Okay, right. So I am not going to discuss elaborately the entire content. That's not necessary. Okay, already I will provide detailed answers, and moreover, I will discuss it in class. Okay, just I will provide how to approach each question, the answer structure. That's all. Okay, right. So the basic rules in paper 2. Okay, how to score the 160 above in paper 2. So basic things you have to follow. You have to provide correct answer. That's important. Okay, so completing the question paper is very, very important. That's compulsory for all paper. Okay, in the UPSC. Okay, so you have to complete the question paper for GS, the optional paper. That's the foremost thing. So you have to increase your answer writing speed. Okay, right. So you have to practice more and more. Okay, if you practice more, then definitely automatically you can will increase your answer writing speed. Right. So completion of question paper, first thing. And in paper 2, you have to give correct answer for a DNA RNA difference or mitosis meiosis or the various laws of heredity. So you have to give correct answer at the same time full answer. Okay. For example, the question, the special types of chromosome. So what are the special types of chromosome? 3 special types. Okay. So polyploidy chromosome or giant chromosome, Lambrusch chromosome, B chromosome. So you have to give the full answer at the same time correct answer. Okay. So that's the first rule you have to follow, giving correct answer. Suppose if you don't know the answer exactly, just leave it. Okay. So don't give any wrong information or if you, okay. So try to give, so please avoid a conceptual error. Okay. That's very, very important in paper 2, since it is technical nature. So most of the faculties, mostly from Agri professors, okay, they will evaluate your papers. Right. So if you give any wrong information or if you are making any conceptual error, definitely they will reduce marks. Okay, they will impact on other answers also. Okay, even if you write good answer, then it will create negative impact. Okay, that's why please avoid a conceptual error. So giving correct answer and a full answer is important, and then wherever possible, give illustration. Okay, so illustration, because here in the notes, okay, in our material, the class notes, I have given elaborately. Okay, for each and every topic, I have given elaborate notes, right? So the definition is important. So you have to give exact definition. Okay, in the elaboration part, in the main body part, you can restrict your answer. You can use your, based on your understanding, you can use your own answer. The illustration is important. For each and every topic, I have given a lot of examples and illustration. Okay, so you have to give illustration plus examples. They are important. Finally, the significance, advantages or disadvantages. Okay, right. So illustration, diagram are important, but the diagrams should be neat and very clear. You should draw a diagram very neatly at the same time scientifically. Okay, so don't use pencil or don't use any vague diagrams. So draw the diagram neatly. So suppose in the question, okay, the typical cell with the suitable diagram. Okay, if you draw the plant cell diagram neatly, then the answer is over. They don't even read the content. Okay, you can write whatever you want. Okay, they just they look at the diagram. Okay, if you draw the diagram scientifically, okay, because the scientifically in the plant cell, the vacuole occupies the central position, not the nucleus. Okay, okay. So in only the animal cell, the nucleus occupies the central position. Okay, so in the plant cells, they have vacuole, a large central vacuole. So the vacuole that occupies the central position, and it pushes the nucleus to the margin of the cell. Okay, the diagram should be it should be clear, neat, at the same time scientifically correct. Okay, at the same time, in the mitosis meiosis cell division, உங்க இஷ்டத்துக்கு நீங்க உள்ளுக்குள்ள chromosomes வந்து போடுவீங்க. Okay, so if you are using an illustration for mitosis and meiosis cell division, so you have to listed correctly. Okay, you have to use the chromosome division correctly. Okay, suppose if you are taking the parent cell with 4 chromosomes, then the daughter cell should contain 2 chromosomes each at the end of meiosis, right? At the end of mitosis, the daughter cells they also contain the same number of chromosome. Okay, so in the chromosome number, you have to mention it correctly. That is a drawing diagram scientifically. Okay, right. So these are the things you have to follow. Okay, so giving correct answer and with a proper illustration and diagrams. For many questions like the various laws of heredity, the genetics questions, you can use the illustration for linkage, crossing over topic, for various laws of heredity, the law of dominance, law of independent assortment. You can use illustration. Okay, right. So these are the basic things you have to follow. Definitely will get more than 160, 170 in paper 2. Okay, right. Okay. For just 2 minutes. Huh? Online students, please wait. Okay. Right. Okay. So first question, differentiate between the following. Okay. Differentiate between the following. Okay. So again, for the difference-based question, so for each and the answer structure, okay, the way of presentation that differs from question to question. But here, for a difference-based question, you must draw the table. You must form a table. Okay. So don't write in a paragraph manner. Okay. So never give paragraph answer for difference-based questions. Okay. So you should form a table. So DNA and RNA. Okay. So what are the difference? So here, so 2 5 marks question, right? So DNA, RNA, and mitosis and meiosis. Okay. Right. So you can use one page for DNA, RNA, and another page for mitosis and meiosis. Okay. Right. So here, the nature of question selection, every panninga. And you have question 1 and 5 are compulsory. Okay. So remaining questions, how to choose the questions? Because yeah, actually in agriculture, regarding agriculture, most of the agri students are okay. So you know that for almost all the questions, okay, except one or two questions, answer for all the questions. Okay. So it's a very challenging one. Okay, how to choose questions correctly, right? How to choose pannalam? Okay. So questions with subparts. Okay. So here, if possible, we can choose question number 4. So there is a high chance that we get maximum marks in question number 4. Okay, the questions are very highly technical. Okay, you can no need to give your elaborate no interpretation. So own judgments just you can give exact answer. Okay, for example, the DNA replication periods, you can give the procedure of DNA replication starting from initiation, elongation, termination. Okay, you can write it in 3 steps. You can give proper subheading along with the diagram. Okay, and then write short notes on the following: 6 influenced, 6 limited, 6 linked characters. So 3 5 marks question, 15 marks 3 pages. So you can use 1 page for 6 linked, 6 influenced, 6 limited. Okay, and then you can polyploidy. Okay, what is polyploidy? Discuss the applications of allo polyploidy. Specific question. You can give exact answer, right? So what are the applications of allo polyploidy with illustration, with the examples? Okay, so there is a maximum chance there. Then question number 6 also. Okay, questions are very specific. You know what to write, right? எப்படி choose பண்ண அப்படினா the questions some questions are open-ended questions. அதான் சொல்றேன். ஓகேவா? So here the directions பார்க்கணும், directives. Okay, the question direction is very specific. Okay, we don't think beyond that. So for example, in the question number 3, define mutation, discuss its role in crop improvement. That's an open question, open-ended question. So you have to define what is mutation, and then for the remaining 2 pages, 2.5 pages, you have to write applications of mutations. That's based on your some students may write very good applications, okay, very good content, or some students may write only 3-4 applications. Okay, so that's if you know the answer very well, and moreover at least you must know about or 6 or 7 applications in a mutation topic, crop improvement, role of mutation in crop improvement. Okay, then only you can get good mark. Okay, so here in the question number 6, the question is very specific. So how will you obtain virus-free stocks using the tissue culture technique? Okay, and then explain its procedure. Okay, then write the functions of cell wall and cell membrane. The question consists of 2 parts. Okay, functions of cell wall and cell membrane. Okay, and then what is embryo culture? Discuss the applications of embryo culture. Okay, so in the question number 6, you have very specific directions. You can follow. Just you can write what is given and what is asked in the question. Moreover, on the answer presentation, you have to give more subheadings. That's important. So here there are a lot of the possibility. We can give a lot of subheadings for the answer. Okay, in the first question, virus-free stocks using tissue culture technique, meristem culture. The answer is meristem culture. Okay, then you have to explain the procedure of meristem culture. Okay, starting from sterilization till the growing of plants. Okay, then write the functions of cell wall and cell membrane. Okay, you can define what is cell wall. Okay, and then the cell wall diagram, the 3 layers, middle lamella. Okay, so here you can draw the cell wall diagram. Okay, and then the functions of cell wall. At least 4 points, that's all. Okay, 4 or 5 points, that's all. And then again a cell membrane. Okay, the functions of cell membrane. What is cell membrane and the functions of cell membrane? So you can give exact answer for these questions. And then finally the embryo culture. Discuss the applications of embryo culture. What is embryo culture? Discuss the applications of embryo culture. Here also again we have for the embryo culture, we have 4-5 applications. The most important one, the embryo rescue technology. Okay, right. So here we first for the 5 minutes, okay, you have to spend time for selection of question. Okay, so in which question we can score maximum marks, right? And question number 2 also, you can select. Okay, in this question, if you are confident, you can take question number 2, 4, and 6. Yeah. Okay. So question number 2 is a very specific question. 3 directives irukku. And question number 3, again 2 in the Bombay, genetically modified croplands and management of insect pests, diseases and weeds. Very specific. Okay. Then male sterility, discuss its types, and limitations of CGMS system. Okay, so the questions are very specific questions. There is a clear-cut directive in each question, so it will be very easy. Okay, right. Right. So DNA and RNA differentiate. Okay. So you have to form a table and the DNA and RNA. So for any difference based question, the first thing you have to do definition. Okay. In agriculture, for all topics, okay, in the introduction part, we can give the definition as an introduction, right? And here also in the introduction part also, first we have to define what is DNA, what is RNA. Okay. எடுத்தோனே டைரக்டா வந்து என்ன பண்ணிராதீங்க. Don't write so DNA is found in the DNA is a genetic material. அது என்ன சொல்லுவீங்க? For example, டைரக்டா வந்து என்னது? DNA is a double standard and RNA is a single standard. Okay. So many students start with a particular point. Okay. So don't do that. So start with the proper definition. First explain what it is. Okay. For any topic, any question, first define the concept. Okay. And tell the examiner that what it is. What is the concept? Okay. Right. So here DNA. So DNA is the universal genetic material. Okay. And RNA is the genetic material. Okay. RNA is the secondary material. And except plant viruses. Okay. So DNA is the genetic material for it's called universal in all cells, living cells, and RNA only in the plant viruses. Okay. Genetic material for plant viruses. It's also genetic material, but not the primary. It is only in the genetic material in the plant viruses. Okay. And you can expand deoxyribonucleic acid and RNA ribonucleic acid. And then you can write the remaining points one by one. It's a double standard, a single standard. Okay. And DNA is produced in the nucleus, synthesis in the nucleus, DNA replication process, and the RNA synthesis from DNA. Okay. You can write the remaining points on just 5 points enough. Since it's a 5 marks question, okay, 1 page, just 5 points. Okay. Not less than 5 points. Okay. You can write up to 8 points DNA RNA difference. Okay. And if possible, you can also draw a diagram. Okay, a small diagram. Okay. And mitosis, meiosis. Okay, mitosis, meiosis. Right. So it's a multiplication division, it's a reduction division. And before that, you have to define what is mitosis, what is meiosis. Okay, direct on another. It's a multiplication division, it's a reduction division. Okay, so don't do that. So first define what is mitosis and what is meiosis. Okay, so here it gives 2 daughter cells and 4 daughter cells. The 2 daughter cells with the same chromosome number and the 4 daughter cells with the half chromosome number. Okay, so here நிறைய பாயிண்ட்ஸ் இருக்கு இல்லையா, more than 10 points. Okay, more than 15 points you can write for mitosis meiosis. You can write at least 8 points. And here you have to give a small illustration for mitosis meiosis. This illustration, that's okay, no need to draw the procedure. Okay, division procedure. So here you can use a small illustration like this. Okay, so at the end of the meiosis, the 2 daughter cells with the same chromosome number. Okay, at the end of the meiosis, okay, the 2n. Sorry. Okay, so mother cell, daughter cells. Okay, the difference. And for the meiosis, you can use the same illustration. Okay. So each cell contains only 1 chromosome at the end of the meiosis. The half chromosome number. Okay, 4 daughter cells with the 4 cells with the single chromosome. Okay. You can use the letter n here, haploid number. It is in diploid condition, it is in haploid condition. Okay, right. And this question again, since a compulsory question are okay, but suppose even this question is asked in the remaining questions, okay, the 2, 3 or 4, it's a very good question to attempt. And the question is split into 2 parts, 5 plus 5. So if you get around 3 marks for 3.5, okay, you can get for DNA RNA difference, you can get around 3.54, and for metastasis you can get around 3.54. So totally you'll get out of 10, 7 or 8 marks. That's advantage. Okay. Suppose for a 10 marks question, maximum you'll get around 5.56. That's all. Okay. If the question is splitted, then there is a chance that we get more marks. Okay. Right. So describe various laws of heredity. Okay, various laws of heredity. So here you have to give introduction. First of all, what is laws of heredity? Okay, don't straight away laws of heredity. The first point, law of dominance, law of segregation, law of inverse assortment. Don't do that. Okay. So first give introduction. Okay, what is laws of heredity? So laws of heredity is given by Mendel through his experiment on pea plant. So give that introduction. Okay. So Mendel experimented with the pea plant, the plant hybridization experiment. Okay. His experiment. So based on his experiment, he proposed 3 laws for the inheritance of the characters. Okay. So those 3 laws are one by one. Okay, you can write law of dominance. Okay, usually 2 laws but actually 3 laws: law of dominance, law of segregation or law of purity of gametes, then law of independent assortment. Okay, and here 10 marks question. Okay, no need to elaborately explain. Okay, just you can define what is law of segregation and you can give the illustration. The 1:2:1 ratio and 3:1 ratio. And for law of independent assortment, no need to use that flow chart. No need to use the chi-square test. That is not necessary. Okay, simply you can use the illustration. Okay, the 4 type of characters. Yellow round. Okay, so what are the 4 type of the plants produced out of the double crossing, double hybrid crossing? Okay, you can define what is independent assortment, the 4 types of the gametes produced in the cross. Okay, and then you can give the ratio. 9:3:3:1 ratio, phenotypic ratio. That's all. Okay, right. So describe various laws of heredity. Then special types of chromosomes. Okay, special types of chromosome. So again, you have to define what is chromosome. Okay, and here for these questions, you can mention the name of scientist. So who coined the term chromosome? Who discovered the chromosome? Okay, the plastids who discovered the plastic? Who found out the plastids? Who coined the term plastids? Those facts are important in paper 2. Okay, while writing genetics part, so you have to mention the scientist name. That's important. Okay, right. So special types of chromosome. First, you can define what is chromosome. Okay, and here in the special types, you can draw 3 diagrams. Okay, for other than the special type of chromosomes, the most important part of the diagrams. Okay, through diagrams only you can explain. So what is a Lamb-Rush chromosome? What is polyploidy chromosome? So what is the B type of chromosome? Okay, so 3 types, special types. So Lamb-Rush, the polyploidy chromosomes are giant chromosomes and the B chromosomes. Okay, you can give a small description of each chromosome. That's all. The answer is over. Okay. And here one more thing for these type of questions, since they are purely technical nature, no need to give any specific conclusion. Okay, so no need to give conclusion. Just you can give the exact answer. That's all. Okay, maybe for the various laws of heredity, in this question, you can think about the conclusion. Like so these laws are further, and the Mendel's experiments are the basis for the further study in the genetics. Okay, so the present day genetics is because of the Mendel's work. Okay, so these laws are universal. Actually, the law of dominance and law of purity of gametes are universal law. Only the law of inverse assortment is only there are some exceptions. Okay, whereas the law of purity of gametes and the law of dominance they are the universal laws, right? And based on the Mendel experiment, many scientists later they discovered and the modern genetics has found its roots in the classical genetics. Okay, now Mendel's class genetics is a classical genetics. Okay, so it laid the foundation for the emergence of the genetics and the further the biotechnology, genetic engineering, plant breeding. So for all these subjects, the base from the classical genetics, Mendel's experiment. Okay, so you can give this point in the two-line conclusion at the end of loss of heredity. Okay, that is also possible. You can improvise your answer. Okay, it's not necessary, but still you can give conclusion for this type of questions, right? Okay. So special types of chromosomes and describe the process of synthesis of mRNA. Okay? Describe the process of synthesis of mRNA. Okay, it's nothing but the transcription process. mRNA on the transcription process. Okay, so first you can define what is mRNA, messenger RNA. Okay, you can write about the mRNA, the introduction part. You can write about mRNA. So it constitutes only 5% of the total RNA, because there are three types of RNA: mRNA, rRNA, and tRNA. The most important RNA in the RNA group is the rRNA, ribosomal RNA. Okay, so mRNA that constitutes 5% of the total RNA. Okay, and the functions, you can write the functions. Okay, what is the function of mRNA, and then you can write the transcription process. Okay, in mRNA on the protein, parts of protein synthesis, transcription and translation. So the transcription process is the mRNA synthesis process. Okay. Write short notes on the following: cell cycle, plastids, aneuploids, test cross. Off page. Okay. So cell cycle, plastids, 2.5 marks. Okay. So here also you can get maximum marks. There's a chance. So what is cell cycle? You can draw the diagram, the cell cycle diagram. Okay, just simply you can just define what is cell cycle, and then you can draw the diagram. That's enough. Okay, and then plastids, draw the diagram and define what is plastids. Okay, if possible, give the types of plastids. Okay, and then aneuploids again. So define aneuploids and give the types, classification in the flowchart manner. Okay, and use the illustration. Okay, 2n minus 1. That's enough. Okay. And any two important applications of aneuploids. Okay, define what is aneuploid, and then give the flow chart, the types of aneuploid. No need to explain. Okay, just give the types. That's enough. It will be the 2n minus 1, 2n plus 1. It will be more than enough. Okay, and then you can give any two applications of aneuploids. Okay. Then a test cross. Okay, so define test cross. Again here, give the illustration. Okay, and the types of test cross. Another types. Okay, monohybrid test cross, double hybrid test cross. So monohybrid test cross ratio 1:1. The double hybrid test cross 1:1:1:1. Okay, கொடுக்கலாம் இல்லையா? புரியுதா புரியலையா? Okay, right. Right. So question number 2, define cell. Okay, so cell is the sexual and functional organization of a particular organism. The define minute and the define cell. How is a plant cell different from animal cell? So here you have to give difference based type. Okay, so plant cell, animal cell difference. Okay, and then typical cell with a suitable diagram. So first you have to draw a neat diagram. Okay, you have to mention all the parts, and then you can the remaining pages you can write the remaining pages you can fill the content. That's enough. Okay, no need to elaborate. Just describe each part of the cell: plastids, mitochondria, important organs. Okay, that's enough. Nucleus. Okay. So B. So second question B, Bombay. What do you understand by genetically modified crop plants? Give an account of their uses in management of insect pests, diseases, and weeds. Okay, each have a specific example, right? Genetically modified crop plants. So, what do you understand by genetically modified crop plants? So, you have to give you a elaborate introduction. Yeah, so first you have to define what is GM crop, genetically modified crop plants, and what are the common examples. Okay, and here the procedure. Okay, half-page or introduction. Okay, and then give an account of the uses and management of insect pests, diseases, and weeds. For insect pests, diseases, and weeds. Okay, for insect pests, we can mention the example of the BT cotton. Okay, what are the BT? Okay, BT cotton, BT brinjal. Okay, you can give all those examples. And the diseases. Okay, diseases category mostly in the virus against virus. Okay, they developed a lot of the GM crops. Okay, you can mention those examples. And the weeds, the soybean, HT soybean, and in India, the GM mustard. Okay, so they are against the weeds. Okay, right. So question number 3. C. Define a male sterility. Discuss its types. Describe the limitations of CGMS system and habits reproduction. Okay, very interesting question. Moreover, we will get maximum marks in this question. So first we can define what is male sterility. Okay. The inability of the pollen grains to fertilize its own ovule. Okay. Male sterility definition. Discuss its types. The types of various types of male sterility. So here a discussion means you have to write, or at least for 20 marks, 4 pages. At least for our 2 pages. Okay, because the answer, the question, the directive is discuss. So you have to discuss various types. Okay, so you have to describe each type. What is CMS? What is CGMS? What is GMS? What is TGMS, PGMS, and the chemical message system, and then transgenic message system. Okay, so there are the various. So here you can in the discuss its types. You can in the first part itself you can give a flow chart. Okay, the types of CMS, male sterility, CMS, GMS, CGMS. போட்டுட்டு GMS நீங்க என்ன பண்ணலாம்? So TGMS and PGMS. Okay, and then transgenic male sterility and chemically induced male sterility. Okay, so you have to describe each type. Then finally, for the last 1 page or half page, you can write the limitations of CGMS. Okay, so what are the limitations of CGMS? And the 3 types of, okay, in the CGMS system, you have to maintain 3 lines, right? A line, B line, R line. Okay, so we have to maintain 3 lines. We need a large area to maintain the 3 lines. Okay, then one more thing, the affected by, influenced by cytoplasmic genes. Okay, right, you can write all those examples, the limitations. 4 or 5 points varum limitations of CGMS. Okay, right. So question number 3, define mutation, discuss its role in crop improvement. Okay, what is mutation? The sudden heritable change. So underlining is very, very important. One more thing. Okay, so in the answer writing, please underline the important keywords. Okay, while writing the base sterility in adu kuda prashana illa. Okay, in the define mutation வரும்போது the sudden heritable change, the word. Okay, so wherever possible underline the keywords. Okay, define mutation, discuss its role in crop improvement. இந்த இடத்துல என்ன பண்ணலாம் அப்படின்னா we can the mutation applications. Actually the question is about applications of mutation, right? So what are the various applications of mutation in the crop improvement in agriculture? Okay, so don't write just list out the points one by one. Okay, for each point give small subheading. Okay, variety improvement, yield improvement, increase of oil content, increasing of grain size. இந்த மாதிரி யூஸ் பண்ற மியூட்டேஷன் வந்து பாத்தீங்கன்னா ஃபார் எக்ஸாம்பிள் இந்த கிரவுண்ட் வெரைட்டிலாம் என்னது there are several groundnut varieties developed by bark bava atomic resistation okay அதுல பாத்தீங்கன்னா அந்த கிரைன் சைஸ்லாம் வந்து என்னது they developed large size the groundnut from the mutation okay mutation variety right so improvement in oil quality oil content improvement in yield improvement in yearly maturity okay those are the applications right the role in crop improvement okay so through mutation we can develop we can produce another the chemically நிறைய application எழுதுறது இல்ல ஓகேவா even we can use the chemically induced male sterility is also a type of mutation we can develop male sterile lines through chemicals இல்லையா okay that's also part of okay one type of mutation so we can develop male sterile lines we can induce the self incompatibility and through bud mutation we can overcome the effect of self incompatibility okay those are the applications okay. So apart from variety development there are a lot of applications in crop improvement so in this point while writing 6-7 points நீங்க எழுதும்போது 8 points okay at least minimum 8 points okay for each point give a small subheading okay don't just list out the points one by one okay அப்ப 8 applications 8 topics மாதிரி இருக்கும் so it will give you extra marks okay right so what is heritability explain the types of heritability and their significance in crop improvement okay so what is heritability define heritability okay the ratio between the phenotypic and the genetic variance to the environmental variance okay right so here you can give the formula okay the heritability formula okay and then explain the types of heritability narrow sense heritability and broad sense heritability then significance in crop improvement just 3-4 points அவ்வளவுதான் வரும் okay significance so based on the heritability ratio we can use this application okay the heritability concept for so here அப்ப என்ன பண்ணணும் அப்படினா நீங்க வந்து என்னது 15 marks 3 pages okay you can use the off page for the heritability definition introduction part off page explain types of heritability so you have to elaborate the broad sense and narrow sense the features of broad sense and features of narrow sense okay and then the significance only 4 or 5 points that's all okay so here we can based on the ratio the number என்ன பண்ணிக்கலாம் heritability we can use it for selection of the parents okay selection of okay deciding the plant breeding program you can decide the parents the inheritability of the characters based on the heritability okay transfer of characters from parents to offspring the ability can be determined by heritability then another important question very specific question describe the gametophytic and sporophytic system of self incompatibility discuss the relevance of essay in crop plants okay so here again you can use 3 pages for the first part relevance of self incompatibility in crop plants வந்து very limited only 4-5 points that's all and major application another we can use the self incompatible plants in the hybrid seed production as a female parent that's all okay right so you can describe elaborately the gametophytic system and the sporophytic system okay you can write all those points because in the gametophytic system we have 8 points more than 7-8 points the features of gametophytic system along with the diagram okay and then sporophytic system the features along with the diagram okay right so question number 4 explain the mechanism of DNA replication in eukaryotes DNA replication mechanism okay a simple question so draw the diagram and the diagram is important okay the replication diagram or replication fork the replication fork diagram is important okay in the diagram itself you can explain everything you can give and the points the pathé முடிஞ்சிரும் அவ்வளவுதான் okay no need to the examiner won't okay they won't read the content okay that means அப்ப என்ன பண்ணணும் நீங்க வந்து you have to give the full information in the diagram okay proper accurate information the Okazaki fragments DNA ligase DNA RNA polymerase sorry DNA polymerase okay DNA ligase DNA polymerase and the action site of action okay everything you have to give in the diagram itself okay write short notes on the following Okay, sex influencer, sex regulatory characters. Just to define what it is. Okay, and then give example. That's enough for each question. What is polyploidy? Briefly discuss the applications of allopolyploidy in crop improvement. Okay, so polyploidy definition, and then here in the introduction part, since it's a 20 marks question, you can elaborate your introduction. Explain பண்ணலாம். Okay, so what is polyploidy? And then you can give the various types of polyploidy in the flow chart, and then you can take allopolyploidy. Okay, right. So in the allopolyploidy applications, you can write elaborately. The 3-4 applications utilizing as a bridging species, and here you have to give both illustration as well as real examples. Okay, so utilizing as a bridging species, for example, the tobacco, nicotina, the example. Okay, so utilized as a bridging species, and then in the allopolyploidy creation of new species. Okay, creation of new species. Triticale. Okay, that example you can give the triticale example, and then widening of genetic base in the mustard example, mustard family. Okay, yeah. Right, section B. So distinguish between monohaploid and polyhaploids. Describe the importance of haploids in crop improvement. So the question is very specific. So monohaploids, polyhaploids. Okay, so just our off page. 2 points, that's enough. Okay, we another 10 marks question. Okay, so remaining 1.5 pages, you can write the importance of haploids. Okay, so off page you can give the difference between monohaploids and polyhaploids. What's the difference? Monohaploids, polyhaploids. எல்லாமே haploids தான். ஓகேவா? So monohaploid means nothing but for a true diploid species, the gametic chromosome number and haploid number are same. Yeah. Okay, for the true diploid species, both the haploids number and gametic chromosome number are same, basic number. Okay, for example, rice is a true diploid species. 2 equal to 24. Yeah. Okay, for rice, 2 equal to 24. So this is the diploid number. Okay, and what is the basic chromosome number for rice? Basic chromosome number, rice, 12. Okay, so the basic chromosome number is 12. So what is the gametic chromosome number? 12. Okay, the haploid number is 12. Okay, so what is haploid number? The gametic chromosome number is called a haploid number. Okay, so for the rice plant, the gametic chromosome number is 12. That is haploid number, and the basic chromosome number is also 12 because for the true diploid species, these two are same. Okay, for the true diploid species, both the gametic chromosome number and haploid number are same. Sorry, basic chromosome number and haploid number are same. In case of wheat, what is the chromosome number? Equal to 42. So what is the basic chromosome number? 7. Okay, so basic chromosome number is 7 because originally the wheat was developed from originated from a diploid species. Okay, so the basic chromosome number is 7. So what is the haploid number? Haploid number means gametic chromosome number. So what is the gametic chromosome number? 21. So here for polyploid species, it differs. Okay, the basic chromosome number and haploid number, it will be there is a difference between a haploid number and a basic chromosome number, right? So this is the difference between monohaploids and polyhaploids. Okay, so monohaploids are nothing but the haploids of diploid species. Okay, haploid plants of the true diploid species, and the polyhaploids are the haploid number of the polyploid species. That's all. Same is the case of cotton, potato. What's the chromosome number for cotton? 52. So cotton 52. Okay, so what's the basic chromosome number? Cotton is a tetraploid species. Okay, so cotton is a tetraploid species. So 52 means yellow round. 13. Okay. So basic chromosome number is 13. But what is the haploid number? 26. Okay. Right. This is the difference. You can here you can define what is monohaploid, what is polyhaploid, and then you can give this illustration along with the example. 2 points, 2-3 points. That's enough. Okay. So describe the importance of haploids in crop improvement. You can give the importance means applications of haploids. So here we can use the haploids for a doubling of chromosome. Okay, to produce true type plants. Okay, then in the potato, the haploid breeding is very easy. Okay, for the polyploid species, haploid breeding is very easy. Okay, potato example, and production of all male plants in the asparagus. Okay, there are several applications of haploids. நீங்க வாங்கிட்டு நோட்ஸ் வாங்கிட்டீங்களா நீங்க? ஓகே, அதுல இருக்கு, எல்லாமே இருக்கு, கொடுத்துருக்கேன். ஓகே, செக் பண்ணிங்க ஆன்சர். ரைட், இது எல்லாமே மெட்டீரியல் எல்லாத்துக்கும் ஆன்சர் இருக்கு. ஓகேவா? So distinguish between the heterochromatin and euchromatin, write the genetic significance of chromosomes. Okay, so distinguish between heterochromatin and euchromatin. The dark heterochromatin and the closely, okay, packed the chromatin, and the euchromatin loosely packed. Okay, right. So you can also possibly draw a diagram, okay, for heterochromatin, euchromatin, and the genetic significance of chromosomes. Again, the significance of chromosomes and the genetic significance of chromosomes. Okay, they are the source of, okay, they are the physical barriers of inheritance. They transfer character from one gene to one species to another species. Okay, so they are the symbol of the inheritance. So then chromosomes. Okay, right. Then define cell theory. Write the features of cell doctrine. Okay, define a cell theory. Features of cell doctrine. Okay, so what is cell theory? The Slade and Swan, they proposed the cell theory in the year. இப்போ இங்க வந்து எப்படி நமக்கு எக்ஸ்ட்ரா பாயிண்ட்ஸ் எப்படி வரும் அப்படின்னா, you have to mention the scientist name along with the year. Okay, so the proposal of the cell theory எப்போ ப்ரொபோஸ் பண்ணாங்க, ஓகேவா? And write the features of cell doctrine. Cell doctrine is nothing but cell principles. நோட்ஸ்ல கொடுத்திருப்பேன். 8 principles, 7 or 8 principles. Okay, there is no much difference between cell theory and cell principles. Actually, cell theory is just 2 points. Okay, all living cells, okay, all the living organisms, they contain the cell. For all living organisms, the basic structural unit of the structural function is called a cell. Okay, all living organisms made up of cells. Okay, and the structural functional unit is called a cell. That is a cell theory. And the cell principle is the elaborate form of cell theory is called a cell principle, cell doctrine. Okay, the cells, they are made up of the carbon, the CHO. Okay, those particles, and the cells, they reproduce through the cell division. All cells, they are derived from the pre-existing cells. Okay, so those are the cell principles, cell doctrine. Okay, again a very specific question. Exact என்ன இதுல வந்து இந்த மாதிரி questions என்ன அப்படினா advantage என்ன அப்படினா, the points are very limited. Okay, அதுக்கு மேல கிடையாது. Cell doctrine only 7 principles. They are the cell doctrine, that's all. Okay, cell theory only just 4 points, that's all. Right, so if you give the full answer, then definitely you will get 7 or 8 marks out of 10. Okay, என்ன பண்ணலாம், applications எழுதுற அப்படினா the mutations applications, there are a lot of applications. We write only 7 or 8. But இங்க வந்து என்னது, the answer is exact answer அவ்வளவுதான். Okay, cell principle னா அது வந்து என்னது, cell doctrine, cell principle னா என்னது, only 7 principles are there. So if you give the correct answer, all the full answer, you will get maximum marks. Right. So why temperature sensitive genetic medicinality is often called as the most efficient system of hybrid cell production. Okay, அதுக்கு ஒரு answer, model answer பார்த்திருப்போம் இல்லையா already. Okay, in the class வந்து என்னது, I will provide a model answer. Okay, I have written a model answer for this question. I will share it in the Telegram group. Okay. Why TGMS is often called as the most efficient system? Actually, இது வந்து purely conceptual. Okay, ஒரு கான்செப்ட் தான். Okay, So if you understand the concept, then you can explain this answer. Okay. So here you can define, first we have to define what is a TGMS. Okay. And you have to give the example illustration. Location A, location B. Okay. So you can here in the TGMS system, you can avoid the disadvantages of the GMS system. Okay, that's a most efficient way, right? Explain the complementary and oppositional hypothesis to explain the mechanism of self-implementation in croplands. So இது வந்து ஒரு சாம்பிள், அதாவது ஜஸ்ட் ஒரு எக்ஸ்பெரிமெண்டல் கொஸ்டின் தான், பாசிபிள். ஓகே, ஏன்னா சோஃபார் நம்ம என்ன பண்ணிருப்போம் அப்படின்னா, வாட் இஸ் செல்ஃப் இன்கம்பேபிலிட்டி, டைப்ஸ் ஆஃப் செல்ஃப் இன்கம்பேபிலிட்டி, அண்ட் இட்ஸ் ரெலவன்ஸ் இன் கிராப் பிரீடிங், ரெலவன்ஸ் இன் அக்ரிகல்ச்சர். ஓகே, அதான் நம்ம காமன் டைப் ஆஃப் கொஸ்டின்ஸ் இன் செல்ஃப் இன்கம்பேபிலிட்டி. இது வந்து ஒரு எக்ஸ்பெரிமெண்டல் கொஸ்டின். We can also expect this kind of questions. The syllabus is only self incompatibility irukku. So what are the mechanisms? There are two hypotheses to explain the mechanism of self incompatibility. One is called complementary, another is called oppositional hypothesis. Just ஒரு 5-6 points இருக்கு அவ்வளவுதான். Just ஒரு 3-4 lines para. Okay, and again very specific question. இதுல என்ன ப்ராப்ளம் அப்படின்னா, அது நிறைய பேருக்கு இது தெரியாது ஆன்சர். But if you know the answer exactly, then you will get maximum marks. And just a theory, if you explain if you give the theory correctly, then you will get maximum marks, right? Then question number 6. How will you obtain a virus-free stocks using tissue culture technique? Explain its procedure. Meristem culture, okay? Meristem culture and the steps in the meristem culture procedure, okay? You can give. Write the functions of cell wall, cell membrane. Embryo culture, applications of embryo culture. Okay, embryo culture applications, the embryo, the most important application, the embryo rescue, then a germplasm conservation. Okay, so germplasm conservation, use panikalam. Then even on the, we can also use the embryo culture for a rudimentary embryo or a very small size embryo. We can use the embryo culture technique. Okay. Question number 7. Discuss the molecular basis of mutation. Again, a very technical question. The point mutation, okay, nonsense mutation, shift mutation. Okay, very important. And the questions in these type of questions recently very popular. Molecular basis of heterosis, molecular basis of mutation. Okay, frame shift mutation. Okay. It's nothing but molecular basis. Okay, you can define what is mutation, and then you can molecular basis nothing but what are the genetic reasons for mutation. That's all. Okay, what are the changes at the gene level to create mutation? Okay, that's molecular basis. What are the changes at the gene level? அதுக்கு நம்ம gene level சொல்லும்போது நம்ம அந்த point mutation, okay, missense mutation எப்படி வந்து அந்த change ஆகுது இல்லையா, the sequence, change in the sequence of the nucleotides. That's all. Frame shift mutation. Okay. So differentiate between the linkage and the crossing over. Did Mendel again is the phenomenon of linkage? Explain. Okay, so you have to define, you have to give the difference between a linkage and crossing over. Okay, and then linkage crossing over கொடுத்ததுக்கு அப்புறம் என்னது, ஏன்னா இந்த linkage crossing over difference ஏன் வருது அப்படின்னா, the question is in a link between a linkage and crossing over, because in the linkage always the two genes they come together. Okay, the two characters they come together. In case of crossing over, என்ன பண்ணும், that will divide, that separate. Okay, the source of genetic variation that comes from crossing over. Okay, so crossing over என்ன பண்ணுது, it creates new characters, new forms of combinations. அதனால தான் we are comparing linkage with the crossing over. Okay, right. So did Mendel recognize the phenomenon of linkage? No, illa. Okay va. So he gave independent assortment theory. அப்ப நீங்க என்ன பண்ணணும், you have to explain with the independent assortment theory. Okay, so Mendel didn't recognize phenomenon of linkage. Okay, because Mendel believed that always another, the segregation of one pair of allele is independent of segregation of another pair of allele, as per the Mendel's laws of independent assortment. Okay, so they always segregate, right? But later Batson and Punnett, okay, 1905 la thane, okay, right, okay, Batson and Punnett, found the phenomenon of linkage in the okay and the Lathyrus oderatus. Okay. You have to give the example of okay there are the two types of linkage, coupling phase, repulsion phase. Okay. So explain abdina, you have to compare it with the Mendel's laws of independent assortment with the linkage. Okay, because in the ratio differ okay, when they tested the double test cross in the double heavy test cross, okay, instead of 1:1:1:1, they got 1:7:7:1. Okay, you have to explain with that fact. So discuss in detail the system of transgenic mensality with a suitable example. Okay, just one type of mensality. So sometimes the questions are very general. Okay, what is defined mensality? What are the types of mensality? And discuss the applications of mensality in crop breeding. That's a general question. Okay, and within the mensality topic, we can expect a specific question like one particular topic. What is transgenic mensality? Okay, explain the procedure. So here you have to define what is transgenic mensality, and then you have to explain with a recent example. That means there are so many techniques available, but the most common example is the use of another Barnes system. Okay, so baseless amylo liquefations. That's the most common system of transgenic mensality. Okay, right. And here discuss in detail automatically, since this is a big question, 20 marks question, after explaining the transmission system, you have to write advantages and disadvantages also. At the end of the question, you have to write advantages and disadvantages. Advantages of TGMS and disadvantages of TGMS. Okay. But you have to write. Question number 8. Discuss the methods of gene transfer to plants. Again, very direct question. Okay. Different methods of gene transfer. The particle gun method. The most common agrobacterium mediated transfer. Okay. So agrobacterium mediated transfer. So in the introduction part, you can write about the genetic engineering technology. Okay. So where we use the gene transfer method. Okay. So you have to give small introduction about the genetic engineering. Okay. So in the genetic engineering technology, we use for the transfer of gene. Okay. We use various methods. Okay. There are several the steps. Okay. One by one. Agrobacterium transfer, particle gun method, electroporation method. Okay. Right. Explain the mechanism of crossing over and its importance. Mechanism of crossing over. Okay. With a diagram. The notes. Okay. Mechanism of crossing over. 4 steps. Okay. So 4 steps in the mechanism of crossing over. Right. Its importance. And then what are chromosomal aberrations? Describe the structural aberration and the genetic significance. Okay. So chromosomal aberration topic both numerical aberrations and structural aberrations. Numerical aberrations are polyploidy, haploidy, aneuploidy, and structural aberrations are deletion, duplication, inversion, translocation. So here give proper illustration. Okay, and the chromosome. Okay, duplication, deletion. Okay, so the illustration is important with the genetic significance. Okay, that's all.